Okay, so what we're going to go over today here, guys, are the lab report expectations, format, okay, things like that. Just a couple of little details about it. First off, lab reports will all be submitted through Google Classroom. Okay, you will get a Google Doc that you will modify and type in and whatever and make it into your lab report. It'll already have your name on it. Okay, so as an example, if I go into Google Classroom right now with my student account, okay, I can go in here and go, okay, I'm in AP period 3, and I can already see that the lab that we're going to work on later in class today is already posted. And if I go into that, my work is right there, and it has my student account name. It'll have your name there instead, so that'll be your file. Now, here's what you need to understand about that. That, and only that file, is the one that I will accept for marking. Okay, you cannot go typing in another program and then submit that file to me. I will not even look at it. Okay, it has to be that Google Doc because I can see everything you do in that Google Doc. I can see the entire history of your editing of that Google Doc. I know that sounds creepy, but it's true. Okay, that basically tells me I can know that you did it. Okay, I mean, if you do it in another program, I don't know whether you got AI to do it or whether you copied it from somebody or what. Okay, so that will be the only file that is accepted. You just turn it in at 8:30 a.m. on the due date. That incidentally is when everything is due. Okay, that lab will be due next week. I made it due on September 17th. Okay. So we'll start the process of doing that later on in class today. What we want to look at first, okay, is format, okay, expectations, etc. So that sheet you have in front of you there, okay, we'll be kind of going through that. You'll want to add in a few things, point values, okay, stuff like that. So first thing is you're going to put in the problem, okay? That'll be blank on your Google Doc. So if I show you what the Google Doc for this looks like, okay, this is what your file will look like, and it just says restate the problem in your own words. Right? So when I put up the lab sheet for this, the problem will be there, and you'll just put it in your own words. There's no marks for the problem. I just need to see that there's something there that's somewhat applicable to what we're doing. Okay? All right, so you'll state it as a question in your own words. So as an example, let's say that we want to do a lab that investigates the effect that light has on plant growth, right? So the problem we would be investigating, what is the effect of light on plant growth? Okay, I state it as a question, like it's Jeopardy, okay, and I write that as my problem. Okay, everybody clear there? All right, first thing that is worth marks in your lab report is the design, okay? The design is out of 5 marks. In the design, you need to identify and explain the variables in this particular lab. So that means you'll need to identify the manipulated variable and explain how it's being manipulated and why. You'll have to identify the responding variable, identify how it's responding, how it's being measured, okay? And then 3 controlled variables, okay? Why are they being controlled? In other words, what effect would they have if you didn't? And how are you controlling them? Okay, so every one of these things gets explained. That's why it's out of 5 marks. 1 for the manipulated, 1 for responding, and 1 for each of the 3, okay, controlled variables that you are doing. Okay, questions there? All right, so in a lab where I'm testing the effect of light on plant growth, what would my manipulated variable be? The amount of light. The amount of light, yeah. Okay, that would be my manipulated variable. So I would, this would be a pretty brief explanation, probably wouldn't get you full marks, okay? I would say, all right, so the manipulated variable is the part of the experiment that's being changed by the experimenter. That part you don't need to write, that's just explanation. That's what I, as the experimenter, am going to change. In this case, it would be the amount of light given to the plants. That's the manipulated variable, because all subjects will receive different amounts of light, and light powers photosynthesis. So I said, here it is, here's the manipulated variable, here's how it's being manipulated. I, as the experimenter, am going to have different subjects get different amounts of light, and here's why. Light powers photosynthesis. Okay? It doesn't need to be super long, okay? You know, it's a sentence or two. Okay, that just does those two things, identifies and explains. Okay, everybody with me so far? Okay, in an experiment of the effect of light on plant growth, what would the responding variable be? The growth. Okay, now, are there a lot of different ways to measure growth? Yeah, and I would probably want to mention how I'm doing that as part of my explanation. Okay, so the responding variable is, in general, whatever changes as a result of your manipulations. Okay, so here, the growth of the plants would be the responding variable because the plants getting more light have more energy for growth. I would add to that, okay, how is it being measured? As an important piece, okay?
Do you mind if I grab a sheet?
Oh, sorry, you didn't get one.
Thank you.
Anybody else missing one?
Appreciate it.
Yeah, oh, sorry about that.
All right.
Okay, so in this case, I would probably say something like, measurements of plant growth will be as follows. The height of the plant will be measured once per week. A picture will be taken of the plant every other day. And the mass of the plant will be recorded every week. Okay, so we'll do the length, the height measurements and the mass measurements on a weekly basis, but we'll take pictures of the plant every other day. Okay, there would be my response, or how I would measure that responding variable. Okay, everybody good with that? Now, your controlled variables. Controlled variables are other things that could affect the responding variable. Okay, lots of other things can affect how much a plant grows. So, knowing that, I need to make sure that those things are controlled or kept the same for all of my plants in this experiment. Otherwise, they're going to throw a monkey wrench into my results. Okay, so as an example, one very easy thing that affects the growth of plants would be water. Should every plant in my experiment get the same amount of water?
Yep.
It absolutely should. Okay, that way I eliminate the effect of water on plants. They all get the same amount. Okay, that's important because if I had, let's say the plant that gets 12 hours of sun got no water at all, that's really going to mess up my results because it's going to die. Okay, and that's probably a plant that would normally have done very well. So in that way, I don't know if my results are affected by the light or the water, and my results are useless. Okay, so controlled variables are things that I need to keep controlled so that I can make sure that the results I get are only due to my manipulations. Okay, so I would identify here, okay, the other factors that could affect it, okay, so we got water in here, same amount of water because water is an important factor in plant growth, so each plant needs the same amount, so it cannot affect the growth rate of the plant. So I've identified it, I said, here's how it would affect the experiment, here's how I'm making sure it doesn't. Okay, and you might even write in there, each plant will get a cup of water a week. Okay, so you can water it at the same time you take its mass and its height, okay, or something like that. All right, another thing. Should they all be the same kind of plant? Yeah, this is hardly a valid experiment if one of the plants is a carrot, one of the plants is a giant sequoia, okay, and one of the plants is like a bean. All right, but those grow at completely different rates. Measurements of height and mass would be completely meaningless, right? So another control variable. They should all be the same type of plant. You might even go as far as to say planted at the same time. Okay, I don't want to have, be using one bean plant that's been alive for 3 months already and plant a seed for another one. Okay, they need to be at the same point in their development and the same species. All right, so same species of plant, different plants grow at different rates. All right, soil. Soil also affects growth. Okay, so they should probably have the same type and same amount of soil and probably growing in the same kind of pot. Okay, so that all of those things that could possibly affect it would be controlled. Okay. Lastly, temperature. Okay, temperature can definitely affect the growth of plants. Okay, higher temperatures to a point are positive, but then after that point, they're going to tail off their growth. Obviously, if it drops below freezing, the plant's not growing anymore. It's dead. Okay. Questions on that? Okay, so all of those are controlled variables. Now, just so you know, again, we want to identify it, and our explanation contains two things. It contains how it's being controlled and why. The why is how it would affect the experiment if you didn't control it. Okay, water affects the growth of plants. We want to make sure they all get the same amount of water, or some plants will grow more than others. Okay, something along those lines. Okay, so this part of your lab is going to be, you know, third of a page, probably something like that. There's a fair amount of typing and explanation to do here. That's the expectation, right? What I don't want to see is the manipulated variable is the light, responding variable is the growth, and the controlled variable, plant, dirt, water. Well, that's going to get you probably 2 out of 5. Okay? You've identified the variables, but you haven't told me anything else. Okay? The explanation part is crucial. Okay? It demonstrates your understanding of the nature of the experiment and the thing that you're testing. Okay? Questions on the design? Okay. Back to controlled variables. Controlled variables generally fall into 2 groups. Okay? Your controlled variables can be procedural and conditional. Sometimes we use the word environmental for conditional. Okay? So procedural controlled variables have to do with how you perform the experiment. So the water, the soil, okay, those things that you're actively doing the same for each plant would be procedural controlled variables. The temperature in the room would be a conditional variable. Okay? You're hoping that it's going to stay around 20 degrees Celsius for the duration of the lab. Okay? What I like to see when people hand in a lab report is a mix. Okay? You're identifying 3 controlled variables. Okay? So at least one needs to be conditional, or at least one needs to be procedural. I don't care which one's the 2 and which one's the 1, but I'd like to see a mix of the 2. Okay? All right. The next thing that comes along is your hypothesis. Now, what I find is coming out of junior high, a lot of people have kind of a misguided impression of what a hypothesis is. Okay? A lot of people tell me it's a guess. It is absolutely 100% not a guess. We don't guess in science. Okay? We come up with ideas and we test them. Okay? We don't guess. So a hypothesis is a statement. Okay? It is stating this is what's happening, this is how it's going to be tested, these will be the results. That way, you can be wrong. Okay? If you just guess, you can get all wishy-washy on whether you were right or wrong at the end, and then it's useless science. It's not even science. It's guesswork. Okay? We want to have a statement, and then at the end of our lab, be able to say, the data goes like this, the data does not support the hypothesis, therefore the hypothesis is rejected, and a new experiment with these modifications will be performed. Okay? Will you lose marks if at the end of an experiment your hypothesis ends up being rejected? Absolutely not. Guys, in real science, real research, most hypotheses are rejected. Okay? And then we change something, we try something else, we try again. Okay? It's always a matter of kind of modifying and going on to something new. Okay? That sort of makes sense there? Okay, so you're never going to lose marks. Because I've had people who are like, you know, they come in and like, Mr. Coderre, can you read my hypothesis over? Which, by the way, I will totally do. Okay? And I read it over, and then they hand in their lab, and their hypothesis is different. I'm like, why did you change your hypothesis? It was wrong. I didn't want to lose marks. Okay? If I told you your hypothesis was good, why would you change it? Okay? Just because at the end you had to reject it, that's fine. You reject it. As long as your conclusion explains why, you're fine. Okay? That's how it goes. All right, so in a hypothesis always contains these 3 words. If, and, then. Okay? It is a deductive statement. Okay? And a hypothesis goes like this. If this is true, and an experiment like this is performed, then the results of this experiment will be as follows, which can be used to do this. Okay? There's no I think in a hypothesis. Okay? This is going to sound really mean, but in a hypothesis, I don't care what you think. Okay? I care what you can prove. That's what science is about. Okay? Our feelings and our thoughts, those have places elsewhere. Okay? I don't know, therapy maybe, I don't know, but not in science. Okay? They don't belong there. Our thoughts and feelings don't go into that because they create bias. If I start thinking, I think this, I'm going to be biased towards that. And then everything I do in that experiment is suspect. Okay? So we never want to bring our thoughts and feelings into a lab report. Okay? It has to be cold, okay, and deductive. Okay, so if we're talking about this experiment with the plants and the growth, okay, I would go something like this. If plants require light for growth, okay, that's the premise I'm working on. That's what the if part always talks about, okay, what I'm testing or what seems to be true, okay, that's what I go with. If plants require light for growth and 5 identical plants are given different amounts of light, so I briefly described my experiment. Okay, I didn't go into detail, that's what the procedure is for. Okay, I just briefly described it. Then the plants that receive the most light will show the most growth. This is a pretty simple hypothesis because it's a pretty simple problem. Okay, but your hypothesis is generally going to be 4 lines. Okay, it's a long sentence. English teachers would probably say it's a run-on sentence. That's fine. Okay, but it's going to be a fairly long sentence where you're telling me these things. If this is true and an experiment like this is run, the results will be as follows, and they can be used to do this. Okay, that's what goes in there. Okay. Does this hypothesis set me up to be right or wrong? Yes, it does. Okay, I am either going to reject this hypothesis or I am going to accept it. I didn't sit on the fence with this. Okay, your hypothesis cannot put you on the fence where you can fall on either side after. Okay, it has to be at the end proven right or wrong. Okay, and they're never like 100% right. They can be accepted, okay, or they can be rejected. All right, questions on the hypothesis part? Okay, so I would say this here. Okay, that's kind of your quick reference for what the 3 parts are about. Okay. The other thing we want to avoid in a lab report as much as possible, but especially in the hypothesis and conclusion, is the use of first person. Okay? I shouldn't see the word I, us, or we in your lab report, but especially in the hypothesis. Okay? Because your procedure, your lab, has to be repeatable by somebody not you. Okay? Science requires validation. It requires repetition. So if I do an experiment and I get certain results, the next step is that guy over there does it and sees if he gets the same results. Okay? If I'm in the hypothesis, I have to be there. Does that create bias? It does. Okay? That's why you see in my hypothesis here, I'm not mentioned, you're not mentioned, only the plants are mentioned. They're kind of an important part of it, so I can talk about them. Okay, make sense? That's going to be probably the hardest habit to break. Okay, is wanting to include I think this, I want to do this. We don't put that in. Okay. All right, procedure. Now, for the procedure here, okay, you're never going to write up the procedure. The procedure is already going to be made for you. You just have to follow. Okay, but know that if you make up a procedure, it has to be detailed. It has to be explicit, and by that I don't mean you're swearing at people in the procedure. Okay? I mean it leaves no doubt as to what should be done in the procedure. Okay? And it is step by step. It is a numbered list that people can follow. Okay? The worst procedures I've ever seen are paragraphs. Okay? I go and do the first thing, and then I come back and I can't remember where in the paragraph I was. Right? If I have step 1, step 2, step 3. I know I just did step 2. I'm looking at step 3 next. It should be a numbered list. Okay? And they are for preparing and performing the experiment. So when I write a procedure, okay, I write it so that anybody could follow it. Okay? Don't take that personally. But I write it so that anybody could follow it and not screw it up. Okay. As a result of, the hypothesis, I forgot to tell you how much that was worth. Sorry, the hypothesis is out of 5 marks. As a result of performing the experiment, you will of course gather data. Those are your observations. Your observations are worth 3 marks. Okay? And I will look at them. If they're way off, you'll lose marks. Okay? Or they're not detailed enough. Okay? That could also cause you to lose marks. Okay? So I'm looking for, certain numbers if it's a lab that involves measurement. Okay? For the lab we're doing tomorrow, okay, it's going to be more qualitative stuff, but some quantitative, temperatures and conductivities and pHs, those would be numbers. Okay? But when we're talking about solubility, you're going to have to write a little bit, like this dissolved easily and the water turned green, something along those. Sometimes you're going to have qualitative observations. Okay? Don't just go, yes. Okay? What if there was like a 20-degree temperature change? You sort soluble. Yep. Okay, that's not really super detailed. Okay? Everyone kind of following me there? It's going to be told to you in the procedure, here's what to record as well. A good procedure always does that. It's not step 1, step 2, step 3. It's also after step 1, record this. Okay? These are the observations you should be making in the lab. So it's important that you read through those. Okay, once you have your observations, then you have to look at, all right, what do these observations mean? That's analysis. Okay? Your analysis is going to look different on every lab, okay? But it's always going to be out of 5 marks. For the lab we're doing tomorrow, the analysis is a chart. You're just going to fill in. Substance A was sodium chloride. Substance B was water. Substance C was this. That's all the analysis you're doing. Okay? Well, that's all you're writing down. You're actually doing a lot more than that. You're looking at, okay, what were the observations for substance A? Okay, it was this, and it was this, and it was this. That fits with that material. That you're doing in your head. All you're writing down is the evidence of that. Oh, substance A is salt. Okay. Everyone with me on that? Okay. If we're doing a biology lab, those observations might be lab diagrams. So you take a picture with your phone through the microscope and then label the thing that you were looking at under the microscope. If we're doing a physics lab, they could be, speed measurements, distance measurements, things like that that we actually took, and graphs that we create from that data. It looks different on every lab. Okay? In the end, what analysis does is takes your data and kind of fits it or doesn't fit it with your hypothesis. Okay? What does this data say? Taking it from facts, numbers, pictures, and turning it into something related to what you were looking for. Okay? All right, for the conclusion. Okay? In a conclusion, you are going to, first off, go back up to the beginning, copy your hypothesis, and paste it. Okay? I want it copied and pasted. Don't bother trying to rewrite it, just copy it and paste it. Okay? The first part of a conclusion is, reminder, here was my hypothesis. The most important part of a conclusion is telling me, here was my data, here's what I interpreted the data to mean, this supports my hypothesis, or this doesn't support the hypothesis. I shouldn't say the hypothesis. All right, so let's say that I did the lab with the plants, okay? And in that lab I had plant number 1 got no light at all, 0 hours of light. Okay? Plant number 2 got 6 hours of light, 18 hours of dark. Plant number 3 got 12 and 12. Plant number 4 got 18 hours of sun, 6 hours of dark. Plant number 5 got 24 hours steady sun, or steady light. My results were as follows. Plant 1 died. Okay? It didn't get any light. It died. Okay? Plant number 2, the 6-hour plant, grew a little. I'm not going to be super specific here because I'm making this up on the fly. Okay? But it grew a little bit. Plant number 3, the 12-hour plant, did quite well, grew quite a bit. Plant number 4, the 18-hour plant, grew the most. Plant number 5 died. the one that was under 24-hour light, died. Okay, so that's my data, okay? I'm looking at that and analyzing, and what I'm getting from that analysis is plants need light. Okay, that was definitely true, because the plant that didn't get any died, and the plant that got a little bit didn't grow as much, and then 12 and 18 did pretty well, but the plant that was under light 24 hours a day died. Does that support my hypothesis? My hypothesis said the plant with the most light will show the most growth. Okay? My hypothesis has to be rejected. It doesn't fit. The data doesn't support that. So in my conclusion, I would talk about that. Okay? I would say those things. So you can see here, okay, as it was found that plants receiving small amounts of light and very large amounts of light showed little growth, or died, okay, it can be concluded that plants require certain amounts of light and darkness to be healthy. Okay? Therefore, the hypothesis is rejected. I could even go into more detail and say, you know, it seems like somewhere like a period of darkness of a minimum of 6 hours is necessary for plants to process things. I could go that far. I could say that because my data says that. Right? That would be a good conclusion. A conclusion should be quite long. Okay? You have to talk about your data, review it briefly, okay, and explain it, and then relate it to your hypothesis. Okay? Conclusion is out of 4 marks. That's it. Okay. Your last thing in a laboratory, okay, is out of 3 marks, and that is your evaluation. In the evaluation, you need to identify and explain 2 sources of error that were within the lab. Okay? You need to identify them, tell me how they affected the lab, and what you could do to fix them in a future experiment. Okay? So let's say that in this lab, I noticed in the lab that the boxes where the light was on for longer got warmer. Okay? I did my best to make sure that the plants that got more light had the same temperature as the other plants, but during my experiment I found that the temperature wasn't perfectly controlled. Okay? Controlled variables are always the best place to look for sources of error, because you almost never perfectly control it. Okay? So I could say in my evaluation then, it was found that plants that were under more light did have elevated environmental temperatures, and this could have affected their growth. Okay? I'm admitting there was something that was observed that I could do nothing about, okay, during the lab that may have affected the data. Okay? I can even say this is how it would have affected the data. Right? How would I fix it in the future? I would make sure that each plant gets its own perfectly climate controlled box. Each one's got an air conditioner attached that keeps it at exactly 20 degrees. Okay? So it could have been that maybe the plant that was under 24 hours of light wasn't about the light. Maybe it just got cooked. Okay? That's something I would have to look at in the future. Okay? And I could say as a second source of error, the plant with no light didn't grow at all. Well, maybe that seed was just a dud. Right? I mean, it's possible I could have put a seed in there that just didn't germinate. It could have had nothing to do with the light. So that could be a source of error. How would I fix that in the future? I'd make sure I had plants that were several weeks old to use. So I would plant more than 5 plants. I would plant 10. Okay? And then make sure, okay, these plants all look about the same. I'm going to put them in the experiment. I know that these are healthy, viable plants, and from here I can be sure of my results. Okay? Sources of error have to be within the procedure. You were told to do it a certain way, you did it that way, and here's some faults in the experiment. You're not going to hurt my feelings by picking them out. They're built that way. They're built for you to find the flaws. Okay? There's always going to be some. What I don't want to see is human error in your list. Okay? Because I get this sometimes. People are like, yeah, like for our lab here. I accidentally got a bunch of substance A in substance B. So, you know, our data's probably off. Yeah, it definitely is off. Why would you tell me you screwed it up and did nothing about it? Your whole lab is suspect now. I can't trust anything in it. Okay, I'm probably going to go back and reevaluate the other parts of your lab after hearing that. Okay, because what you basically told me is, yeah, I screwed up and I was too lazy to check it. Okay, no, that's not how we do it. If you screw up, that's fine. It happens. Okay, there's probably going to be a couple of groups that will accidentally get some of A and B. That happens. But then you go, okay, I can't use that one. We'll do all the rest. We'll come back and we'll do B again. Okay, you fix it in the lab. Never list human error as a source of error in the evaluation, because human error can be corrected, and it should be corrected when it happens. Okay? All right, so a whole lab is out of 30 marks. Okay? Labs go in your coursework part of your grade, which is worth 20% of your final grade. By the end of the semester, you'll have 12 to 15 things in that category of your marks. Okay, so every one of the labs you do is a little over 1% of your final grade. Okay? Questions on report format and expectations? Okay, last note on these. You can always come in and have me look these over. Okay, so if you go home tomorrow after having done the lab and you start working on the lab report, okay, and you've got most of it done, if you want to come in and see me before school or at scheduled help or at lunch or any of my help times, okay, if you want to come in and say, Mr. Coderre, can you look over my lab, please? I will do that, and I will give you feedback. Okay? Sometimes that feedback is, holy smoke, this is amazing, change nothing and turn it in. Okay? And sometimes it'll be like, you forgot to explain your controlled variable number 1, or your hypothesis doesn't have an and part. It's just little things, details that would cost you a mark or two along the way. I'll catch those, and I'll give you suggestions on how to fix them. I'm not going to tell you the right answers, okay? But if I see something, we'll work it through, and then you get that feedback and go back and change it, and you do better, okay? And you learn. It's totally up to you. You don't have to do that, but I will. Yes. Okay? Questions on that? All right, I'm going to give you about a 2 or 3 minute break here within the classroom before we start doing the pre-lab stuff. Okay, so chat amongst yourselves.